Alright guys, so this is the last lab of the semester. We're going to be discussing echinoderms and chordates. We previously talked about protostomes, which that's where the blastopore forms the mouth before the anus. Now we're going to talk about deuterostomes, which is where the anus forms in the blastopore before the mouth. So we're into chordata. So these are all chordates. They have these 5 chordate traits that we'll talk about soon. But to start, we're going to discuss the phylum Echinodermata. So there are 4 basic characteristics in this phylum that you'll need to be familiar with. This phylum consists of things like sea urchins, sand dollars, sea cucumbers, and sea stars. Here we have a little sea urchin. You can see its calcite skeleton. It has these little bone-like spines, like bone-like plates that stick out of the body, and they are made out of calcium carbonate. We also have these structures that are called pedicellare, and they're used for self-defense in these organisms. The second characteristic that you'll need to know is the water vascular system. So everything in this phylum has a water vascular system. On your phylogenetic tree, you may see it abbreviated as WVS. But this water vascular system aids these organisms in feeding, respiring, even moving around, or just attaching to structures. And there are 7 parts of this vascular system that you will need to be familiar with. Next, we have some mutable connective tissue. So it's easily can change stiffness of the tissue. For example, it can take its stomach, which is stiff on the inside, and then change it into a softer tissue and invert it outside of its body. So this is a starfish that has its stomach outside of its body. Really interesting. It's also a defense mechanism. And the 4th characteristic is that these guys are what's called pentaradially symmetric. So think of radial symmetry where you can kind of cut it every way that you want, but it's pentaradial, so it's kind of upon one of those 5 axes. So you can cut it, long ways, kind of down here, kind of like this, in order for it to be completely symmetric. So the adults are pentaradial or radial, and then the larvae are bilateral, so you would cut this larvae right down the middle. So here are the parts of a starfish, for example. This might be a question just asking you to explain different parts. So you can also watch this video that's attached. For reproduction in these organisms, primarily sexual, but you may have heard that if a starfish loses an appendage, it can reproduce asexually. So that would be an example of fragmentation. It can regenerate its arms or spines or other organs and tissues. So there are 5 classes within Echinodermata. So we have Crinoidea, Asteroidea, which are our sea stars, Ophiroidea, Echinoidea, and Holothuriidea. So we'll go through each one. So Echinoidea, these are sea urchins and sand dollars. They're just globe-like. They don't really have any arms or anything. And the tube feet usually extend around the sides of the body. So make sure in your lab manual you're drawing these things. List the 7 parts of the water vascular system, starting with the madreporite, and you can just keep listing. In the sea urchin dissection, you're going to need to be able to identify some parts. For example, the aboral surface, that's the side with the anus. So that's not the side that they eat on. If I'm asking for the oral surface, that's the side with the mouth, right? Spines are pretty obvious. Madreporite is the opening near the mouth right here, for example. Internal anatomy, so we have gonads, esophagus, Aristotle's lantern, which is the bigger, like ring-like structure on the inside. Holothuriidea, these are our sea cucumbers. They are fleshy, sausage-shaped. They have an endoskeleton that's just some ossicles, right, which are to differentiate light and darkness. They have tube feet, or they don't have them at all. And then their madreporite is on the inside. So if I asked you where the madreporite is located, you would say internally, right? So the class Ophiroidea, so these are brittle stars, basket stars. They look like this. They have 5 flexible arms radiating from this central disc here. The madrepore is located on the oral surface this time. So sometimes you'll see the madrepore on the aboral surface, sometimes on the oral surface. Tube feet lack suckers, so they don't really have those suckers to stay attached to things. They're more mobile. So how would you describe these animals? How do they differ from Asteroidea? So the sea stars, one of the biggest differences is that they do not have those tube feet, those suckers. And then finally, Asteroidea. So these are our sea stars. They have 5 arms radiating from that central disc. They are pentaradial. The madrepore is on the aboral side, so that's another difference between the other group. And they do have tube feet on the oral surface. They are considered predators or scavengers, believe it or not. And then some of them, like I mentioned before, have that irreversible stomach, so they can digest food externally. So pedicellare, remember, are for eating and self-defense, right? And then comparing the morphology of a sea star to a sea urchin, you can kind of go back and look at the anatomy and do that. And then this class here, so class Crinoidea, these are feather stars and sea lilies. They're really interesting looking. They have feather-like arms, and they'll have more than 5 arms in this case too. They have tentacle-like tube feet. The anus is actually on the oral surface this time, so that's another strange difference in this class. They have a sessile stalk phase where they're non-mobile and they're just kind of there. And then no spines and no madrepore in this class either. So for this part of your lab manual, you can just kind of look at these pictures and just make note of their structures and how they're different than the other groups. So now we're going to get into phylum Chordata. These are lancelets, tunicates, and vertebrates. So there are 5 chordate traits that you are going to need to be familiar with. The first one is a notochord, which provides skeletal support. There's a pharyngeal gill slits. So even, as humans, when we're in the embryonic development, we also have gills at some point, but we do lose them. So these just help us strain food from water particles, right? We have a dorsal hollow nerve cord, which is going to develop into the central nervous system, for example, your brain and other parts of your spinal cord. Postanal tail, which humans also do have during development, but they do lose it. So in aquatic species, they retain the tail, and it's usually used for propulsion or movement in the water. And then finally, an endostyle, which is a mucus-secreting tissue that traps food inside of the pharynx. So organisms in this phylum, they are deuterostomes, they are bilaterally symmetric, they're triploblastic, so they have all 3 of those tissue layers. They're eucoelomates, so they have a true coelom, and reproduction is primary sexual. There is some asexual reproduction in our tunicates. So now we're going to look at the subphylum Cephalochordata. So these are the lancelets. You'll notice here that these are marine filter feeders, and what's really interesting about these is that they exhibit all 5 chordate traits. So they have all 5 traits. They're visible. You can see them here. So I'll ask you to list 4 defining characteristics of chordates that you could see in this group, okay? And then which chordate trait is missing. They in the slide, see if you can identify the traits and then see what you think is missing from that slide. Okay. So then we have subphylum Tunicata. These are sea squirts. They're pretty interesting. If you're ever on a boat dock, you would be able to see these guys sticking to the side of the boat dock. They're sessile. You can actually squirt the water out. That's why they're called sea squirts. They are filter feeders. The adults only have the pharyngeal gill slits. They don't have any of the other 5 chordate traits retained. The larvae, however, will have all 5. So See if you can list two differences between the larval stages and the adult tunicates. And then subphylum Vertebrata, so these are lampreys, sharks, fish, birds, humans, everything else. In addition to those 5 chordate traits, we're also going to have a neural crest, which forms skeletal elements like the bones. There's pronounced cephalization, so remember cephalization is when you have those sensory parts all concentrated in the same area. We also have a vertebral column in this group, so skeleton of bone, some cartilage as well, and there's also a closed circulatory system. So now we're going to go through 6 classes and then 1 order that you'll need to be familiar with. So the first order, these are all of the lampreys. So these are really interesting organisms. They're petromyzoniforms. So lampreys, they're some of the earliest ones. They look pretty primitive. They're jawless, so they do not have any jaw of some kind. They just have this like one sucker mouth. Their endoskeletons made out of cartilage, no scales. They don't have any paired fins like in other fish. So what do these early vertebrates lack? Lots of different things. Primarily a jaw, scales, no fins, right? Class Chondrichthyes, these are sharks in addition to skates and rays. So they have cartilage. They have a very flexible endoskeleton that's made out of this cartilage. They do have jaws with paired fins, so you'll see a dorsal and ventral fin. They're viviparous, so they have live young, but they can also be oviparous, meaning that they lay eggs. And then they have what's called a lateral line system that allows them to sense all kinds of things. So describe the skeletal makeup, and then one other characteristic that unites the animals in this class. So definitely that lateral line skeletal makeup is out of cartilage, right? So their endoskeletons made out of cartilage. Class Actinopterygiae. So these are rayed fin, bony fishes. These are your typical fish that you see. They do have paired fins. Their endoskeleton is made out of bone, like I mentioned before. They have scales, they have a jaw, they have a lateral line. They also have a swim bladder that's used for buoyancy, so it allows them to kind of float. And most of these guys are oviparous, so they push their eggs outside and they're fertilized that way. So what's the primary component that makes up the endoskeleton? Bone, right? So this one's made out of bone in comparison to cartilage. And then we have Sarcopterygiae. So these are lobed fin bony fishes. These are really early fishes. I actually met the guy who discovered the species, the Tiktaalik rosier. This was one of the first fish with feet, or so we think. And there are 8 species that are still alive today, like the coelacanth, and then the lungfish as well. If you want to see something super ugly, look that up. So how are the animals in Sarcopterygiae different than Actinopterygiae? Well, definitely what they're made out of. The fins are super different as well. And then finally we have these classes, so Amphibia, Reptilia, and Aves. So our amphibians, they have 4 legs primarily, spend part of their lives in the water, have super smooth skin. And the biggest difference between the amphibians and the reptilians have an amniotic egg, and they're going to lay their eggs on land. Amphibians typically need water to lay their eggs. And then finally, aves, so these are our birds. They're actually more closely related to dinosaurs than anything else. Their feathers are modified scales, so that's the connection there. They have wings, a beak, and they lay eggs in order to reproduce. So you can look up the 4 stages of metamorphosis in amphibia. You can also describe a similar characteristic between the 2 classes. So the fact that they do lay eggs, but note how Some of them lay eggs on land and the others are in the water, right? And then finally, class Mammalia. So there's a couple different mammalian traits that you can be familiar with. For example, mammary glands in mammals are used to make milk. We have mammalian hair in some shape or form, so it's either fur, whiskers, sometimes even horns is modified mammalian hair. There are 3 middle ear ossicles. We have 4 chambered heart, endothermic, and they have a cecum, which we'll see on the next couple slides. So I usually have these memes here because we typically dissect a rat, and it's pretty hard to be okay with. But you can talk about the 2 characteristics that make a mouse or a rat an animal. There are lots of different a mammal, I'm sorry. There are lots of different mammalian traits that the rat or mouse would have. Definitely the hair. On this slide, they don't have a cecum. That's what they're lacking. But you can label these parts here. This would help you as well. This is another one. So be able to identify these parts. So if we give you this sort of image on the practical, make sure that you can differentiate these parts. So please reach out if you have any questions. I'm always here to answer them. And best of luck on the lab practical next week.